Okay. Hi everyone. So before I start my talk, I would like to thank the committee for the invitation. It's really, really nice to be here. Especially at that time of the year. I'm coming from northern Norway, so it's almost the winter up there. It's getting snowy and really cold, so I really appreciate getting a bit of sun and warmth this week. So thank you. I don't know if the slideshow will start. Yeah. So today I will tell you a bit about an Arctic breeding bird, the Bronyx guillemot. It's really a case study about a bird breeding in the Arctic and how the population responds or has responded to changes in the environment. And it won't be really about phenotypic response, unfortunately. I'm sorry for that. When I accepted to come some months ago, I really expected to have some results ready about phenotypic plasticity in relation to different factors. Those results are clearly not ready yet, so my talk will mainly focus on population response. I hope it will be okay. At least I put some nice pictures. So, just a few words about Bronyx guillemot. I won't get into details about the Bronyx guillemot biology. It's a long-lived seabird, like almost all seabirds. Delayed first breeding, population growth rate, highly sensitive to adult survival, a bird that can skip reproduction, and all these kind of classical features for seabirds. It's really a bird breeding in the Arctic. There is no Bronyx guillemot breeding in temperate environments, or really an Arctic breeding seabird. And the Svalbard population, it's close to 1 million breeding pairs. It's a least concerned species. Basically, there is a lot of Bronyx guillemot. It's doing relatively well in many places on Earth, in Canada, in the States. It's okay in Greenland. It's doing relatively okay. So least concerned species, but of course, if you look into more specific, like regional scales, status can be different. And it's a vulnerable species in Norway, based on the red list, Norwegian red list, and near threatened species in Svalbard, which is the real Arctic part of Norway. And just to show some numbers, to show how important this Svalbard population actually is in terms of European stock. In Europe, we estimate that there is 2.5 million of Bronyx guillemot, breeding Bronyx guillemot, and the Svalbard population itself is from 1/3 to half of that population. So of course, it's from the conservation or management perspective, it's a very important population, of course, in terms of number of individuals, just to emphasize that. In Svalbard, which is composed of several islands, we have quite good data actually on 7 colonies, monitoring data. So basically since 1988, there has been people going to those colonies, counting birds in the same places year after year, and we use that as a proxy of colony size, at least colony attendance, which is correlated to number of breeding birds. And this is what the data showed us. So the raw data, just standardized, put on the same scale, and you can see that it's quite obvious. It's one color, one line per colony. And since about 15 years, there is a drastic decline of about 3, 4, 5%, depending on what kind of model actually, or what kind of estimate you consider, but quite a drastic decline, and quite clear since about 15 years. If you run very simple stochastic models, basically just to say, okay, what will happen if it continue in that direction with the same kind of variation, what will happen for the population? And if you want to quantify the risk of extinction, or more exactly quasi-extinction, basically what you have to remember from this figure is that it goes fast. In 50 years, if nothing changes, we have 1 chance out of 2, or 43% of chance that population will be strongly extirpated. In 100 years, if nothing changes, which is of course a really, really strong and unlikely assumption, population will be extirpated and almost quasi-extinct. So just to say that it goes fast. This decline goes really fast. Interestingly, if you consider the onset of decline, when had this population, these colonies started to decline, there is no difference between all those colonies. They all have started to decline around exactly the same period. Some noise, of course, the confidence interval is quite wide for some colonies. We're not talking about hundreds of data, but it's only 20, 25, 30 points per colony, even less for some. But still, they all have started to decline around the mid-90s, 96, 97, 98. If you look at the rate of decline, same conclusion, or more or less same conclusion, they're all declining at the same speed, at the same rate, all but one, this one, Fuglehuken, which is located, I don't know if I can show it, this one here, on the this western island. It's declining a bit more faster than the other colonies, and we have absolutely no explanation for that. We don't know. It's maybe simply an artifact. There is one data point in time series that is a bit lower than the other. We don't really know. It's no good biological explanation. We don't even know if there is any biological meaning behind this faster decline. But I think the main conclusion is that most of these colonies are declining at the same speed and are declining since the same date. And if I go back just to this, and of course the important thing here is that it tells us a bit about the mechanism, the environmental driver behind this decline. We're not talking about something happening at a very small geographical scale. We're talking about something that happened at a quite large scale to affect all those colonies in a similar way. But I'll come back to this in a few minutes. Okay, colonies are declining. Population in Svalbard is declining quite rapidly. Now we want to understand why, which means to understand why from a demographic perspective. What are the drivers, mechanistic demographic drivers to explain this decline? And then, of course, what are the environmental drivers behind this decline? In terms of demography, we don't have that much data, actually. Time series are quite young for most of the parameters. If we're talking about breeding propensity, breeding success, juvenile survival, we're getting there. But for now, we don't have anything really available. The only data we have, it's data on adult survival from one colony, which is this Bjørnøya colony, the island just south of Spitsbergen. Spitsbergen is this big island, Bjørnøya this one, and we have quite a good data set on recapture of ringed individuals since '96. And these results are the result of a master thesis from Julie Fleur. She has done her master in collaboration with me and people in the CNRS in France. And Julie found out that, okay, this showed the annual estimates of adult survival. And what this modeling tells us is that, survival was roughly constant, very, very few variation from 96 to 95, 96, 7. We can't really tell which year exactly survival started to fluctuate, but it's in the mid-90s, let's say. After that period, survival started to decline, to vary quite significantly and to decline on average, and a decline of 9 to 12% with quite significant fluctuation. And for this kind of species, as many of you know, such a decline in adult survival, which is the parameter of which has the highest impact on population growth rate, it's an enormous decline, 9 to 12%. It's a huge decline. It must have an impact on population dynamic. Even if it's obvious, we wanted to confirm that, so we ran some, again, simple demographic models. Not exactly that one actually, but very similar. So we consider delayed first breeding at age 5, actually, in this species, juvenile survival, adult survival, breeding propensity, birds can switch from a breeding state to a non-breeding state, and so on. I won't get into the detail. We don't really care right now. And I told you some minutes ago that we don't really know the average values of breeding success, breeding propensity, and all those demographic parameters. So we just simulated this model. We estimated growth rate with many different kind of values for those parameters. And basically, the conclusion of that is that whatever the parameters you consider here, a decline of 9 to 12% in the average adult survival is not sustainable to the population. It's not a surprise, at least we quantify that, we confirm that, but this decline we observe in adult survival has to play a role, must explain at least partly the decline in population size observed in Guillemot. We do have data on body mass, not a very long series, but at least we do have some good data since 2005 in that colony. It's a colony that where I'm working quite intensively since a few years, here in this fjord. And if we look at body mass at capture, when we capture bird, we just weigh it, and if you adjust this mass for the capture date and for other potential confounding factors, we find this kind of results. So again, body mass is declining. Both for male and female, there is no difference in the slope. If we consider individual into account, It's not a lot of repeated measures, but we do have some, and if we consider that, if we take that into account, there is no difference in this slope between individuals, so it seems that all individuals do respond in the same way. But quite a clear decline in the body mass of bird. If you adjust for body size, so let's say if you get an index of body condition at the same slope, and birds have lost on average 5 grams per year. It does not seem to be a lot. Does it have any biological meaning? It can probably have, because over 10 years, we're talking about 50, 60 grams less now than 10 years ago. So it can have an effect on reproduction of birds. We're working on that currently. So okay, we know that adult survival has declined at the same period than population size has. It played a role, obviously, in the population decline. Body mass is declining too, which could play a role as well through an effect on reproduction. Now we want to understand, okay, what has changed in the environment? How could we explain all those changes in the population? And the first step is to know what is the environment that your colony is using. If you don't know where the birds are, what they're doing, basically you can't really answer that question. And seabirds, by definition, are birds that spend most, if not all of the time at sea, except when they come ashore to lay their egg and feed their chick. You don't see them, they're just at sea feeding. So we have to find a way to know where those birds are going to. And Thomas this morning mentioned different kind of loggers, and of course in the seabird world, many kind of loggers are just exploding right now. Many people use loggers to answer to those questions. And this is what we have done some years ago. We deployed some what is called GLS logger, geolocator loggers, which I won't get into detail here neither, but they use a light intensity to basically to estimate the latitude and longitude of the bird once or twice a day. And they can last more than a year, so you can basically track a bird from its breeding ground to its wintering ground and so on for ideally a few years. So with this kind of new technology, we're able to identify the wintering ground of the bird. This is one example here. This is birds from Bjørnøya here that overwinter here in south of Iceland. Birds from Spitsbergen, so the bigger island just above, that tend to go more to the southwestern corner of Greenland, but roughly they use this North Atlantic area. So now we are able to look at the environment in those places and see if variation in this environment is linked to variation in the population. And I will mention this subpolar gyre. The subpolar gyre corresponds to some movement of cold polar water here in mainly Northeast Atlantic. And this gyre can be more or less strong or more or less weak. A weak gyre means that there will be a large inflow of warm tropical water. So when the gyre is weak, the sea temperature here will be higher, the salinity will be higher, and this is associated to many changes in the oceanographic ecosystem. Well, it's in Norwegian here, I'm sorry, but basically it shows that the red line, that the gyre index, and it's correlated to sea temperature, sea level, plankton density, copepod density, and so on. And you see that in the mid-90s, this gyre has clearly shifted. There has been a large-scale oceanographic shift in this North Atlantic area. Other figures to really stress that out. The gyre index in red, so when it's high here, that means a weak gyre, warm water, and it's correlated to plankton productivity, primary productivity. So warm water, high primary productivity. Copepod density, it's one specific species of copepod, Calanus finmarchicus. And here, just look at the scale, it's reversed. So when the gyre is weakening, Calanus density, copepod density is getting lower and lower. Relation at the fish level, again, relation between the gyre index and the fish abundance. And you can even go to the whale level. There is some correlation between the gyre index and pilot whale abundance. So this shift or changes in the subpolar gyre has had many large-scale consequences at different levels of the ecosystem. And if you look at correlation between this gyre index and colony size, it's highly and strongly correlated. And even if you detrend those time series relation still holds. So we have quite good correlation. Of course, it's only correlation, but still, between this gyre index and the colony size fluctuations. It works even if you don't consider any lag, but it works even better if you consider a lag of 5 years. 5 years being the age at which juvenile start to recruit, so it makes sense. There could have been an impact of the gyre through different kind of, through plankton density, through fish availability and so on, up to juvenile survival and recruitment. That's how we can interpret those things, those results. If we look at survival, same kind of relationship. Of course, a lot of variation around this regression line, but the gyre itself explains 20% of the variation in adult survival rate. So it seems to have played a role. And if you just draw a line here, you can see that when the gyre is low, when the North Atlantic was warm, survival of adults was low. When the gyre is strong, when the North Atlantic was cold, survival was high. Body mass and subpolar gyre index, no clear result up to now. It's something I'm working on currently. There seems to be some association, but of course we're talking about a very short time series, so it's still not very clear. There seems to be some association between, again, this subpolar gyre index and body mass of birds, but a work in progress. I just want to stress a bit the role of this little guy here, this copepod species. The abundance of this copepod has really declined since the mid-90s. And according to other papers and other authors, it seems that this copepod is really tightly linked to many different aspects of seabird life history in the North Atlantic. Here it's an example of breeding success in relation to copepod, this copepod. And this author, Morten Frederiksen, a really, really famous and good seabird ecologist, he really considered that this copepod species is a keystone in the North Atlantic marine environment. He compared the role of this species to the role of Antarctic krill in the Antarctic Ocean, so really like a keystone species. So we'll see if other studies confirm that role, but if this is true, considering the decline of that species in the mid-90s, it's not necessarily surprising that now we see consequences at the seabird level and a decline in some populations. So just some first conclusion to wrap up a bit all those things. There seems to be some link. Again, it's correlational, no experiment as we have seen this morning. It's not really doable to set up any experiment to manipulate those things with seabirds, but at least some good correlation between subpolar gyres, some environmental features, and population size. A shift in the mid-90s, shift as well or changes in the mid-90s for adult survival, which seems to have played a role. The lag we observe between the gyre and population size suggests the role of juvenile survival as well. And we do observe a decline in body mass, which could be linked to changes in reproduction as well. So here it's many question marks, of course. We have to confirm many things. But it seems that the relation between the environment, how environmental change has affected the population, has been through many different parameters, which have to be taken into account to really get a good understanding of what is going on. Just how much time? Okay. And here, of course, the diet. It's always a black box, or not always, very often a black box when you study seabird. You have link between global index, this kind of index, and population, but you don't know a lot about the diet and the food availability. So this is something we're trying to look into more details. Again, work in progress, and I hope to get more results linking this gyre, the food of guillemot, and their population development. Just a few more results or comments or notes in this topic. We have observed other changes in regards to Brunix guillemot population, and one of these changes has been the increase in the tick prevalence, the seabird tick, so an external parasite, and increase in relation to winter temperature. This tick, the seabird tick, Ixodes uriae, has been observed in only one places in Svalbard and only one species. So we're not talking about something that is really spread out all over the archipelago. There has been a lot of studies on seabirds since decades in those places. Many different teams, many different people, program, research going on, many different species and colonies studied. It's only in that guillemot colony that we ever found any tick. And the first one has been found in 2007, and since that time, it seemed that slowly the tick prevalence is increasing. We're talking about a few tick maximum per bird. In some cases, we found once one bird with 200 and something ticks. This bird has died. But otherwise, the birds we capture, they have 1, 2, 3, 5 ticks, 10 maximum. So it's very likely not a huge pressure for the birds, at least no direct important impact on the bird health. But interestingly, we found with this impressive data set of 6 points, a very good relation between the winter temperature, either if you consider the average of the mean or the average of the lowest temperature, that's the same relation between winter temperature and the number of birds of guillemot that have ticks in the following winter. If we add the point from the last season, it's not underlined, but it's not too far, and the relation does not really change. So it's small data set, small data series, it's a correlation again, but quite good link between winter temperature and tick prevalence. And this threshold here of -12, -13 degrees, it makes sense based on what we know about tick. It has been published that tick do not survive temperature below -12 degrees, except the eggs, but otherwise they just die. So it makes sense that below those temperature, or around those temperatures, there are very low ticks. But again, there is a relation between temperature and tick prevalence, but no relation for now on breeding success, on bird body condition, on any other threat. So yes, ticks may increase, but the effect on the bird is not yet observed. If I have time, we have still a few minutes. I would like to stress a bit the wind condition in that place. And it's going to start to be very speculative, or at least based on very soft grounds. It was before as well, but it's even worse now. Glaucous gull, we know, we observe that. That's the main predator of Brunic guillemot in that place and in other places in the Arctic. That's really the main one. It can eat a lot of eggs and chicks and so on, and have quite a large impact on the colony. And we know that the wind characteristics, and in particular wind speed, can affect the predation rate and the success of the predation of Glaucous gull. And the wind has changed, or is changing, in Svalbard. An example here, the wind in one fjord where I'm working on guillemot in that specific fjord, and the wind direction here is changing. It was more from the east 20 years ago, and now it's more from the south. So there is a potential here that wind is changing with potential consequence on predation, pressure, and then bird breeding success. No evidence yet, we're working on that, but we do have the feeling in the field that predation by Glaucous gull is increasing, and he's having more and more impact on bird breeding success. So there is something here that may be, with huge question mark, to changes in wind speed or direction and global change. But again, big question mark. And the main take-home message, let's say, of all of that is to say that yes, we have potentially identified changes in the environment that could explain decline of this guy here, but there may be, or there will be potentially other stressors that will play a role in the system as well. So my first conclusion was to say, okay, there is many traits, many parameters involved in this decline. There may be several stressors as well. And I could have added actually one arrow here with pollutants. There is some specific pollutants that are really increasing currently in Svalbard, the fluorinated pollutants, and they could affect, or we know they can affect the hormonal system of the bird with potential consequence on reproduction as well. So it could be another stressor here. So several parameters involved, and we have to take that into account to understand what's going on. Several stressors involved, and again, we have to take that into account to get a good understanding. And just a few words that can make the link with the session, actually, phenotypic response, is this is the way we have to go, as Thomas this morning and other speakers have stressed. We have to look at what's going on at the individual level. It's pretty similar to a slide from Thomas, who was mainly talking about feeding ecology, but it's the same conclusion for whatever the trait you consider. If individuals do respond in a different way to the same environmental variation, what you see at the population level can be completely biased and wrong. So you have to understand what's going on at the individual level to really get a good picture of what's going on at the population level. And that's exactly what we're trying to to move forward this kind of question. So I think I will stop here. It's probably time. So just I skip these slides. And I would like to thank all my colleagues and students involved in this work, and to thank you all for your attention. Thank you.